Mic off.
Welcome to the conference session called This is Your Brain on Chemo Drugs. Thank you for joining us. My name is Julie Powers, and I'm the Senior Director of Patient Advocacy, and I'll be moderating the presentation today. As we get started, we recognize the generosity of our corporate sponsors and our patients, families, and caregivers who have made financial contributions to the foundation in support of today's conference. Immediately following the presentation, there will be a question and answer session. Please feel free to submit questions at any time during the presentation from the Q&A icon in the engagement panel on your right. To submit a question or comment, type your question in the Q&A text box window, and when you finish typing your question, hit enter. We will do our best to get to all questions. When asking questions, I respectfully ask that you do two things to help me manage the incoming questions. First, submit your entire question all at the same time. Second, please do not share your private health information in your question. Our speaker cannot answer any specific questions related to your health care. Also during the session, we will open and close polling questions from the audience to respond in real time using the polling feature on the right-hand engagement panel of the session. Please click on the icon in the panel now and then you can navigate between that and the Q&A window. The poll will only be open for the first 5 minutes of the session, and we ask that you take that poll as soon as possible in order to help us with future planning. Your responses will help us help healthcare professionals with their learning as they learn more about our patients who have bone marrow failure diseases. With that said, today's specialist is Dr. Dietrich, who is the director of the Cancer and Neurotoxicity Clinic and Brain Repair Research Program at the MGH Cancer Center and is associate professor of neurology at Harvard Medical School. His clinical interests are the management of patients with brain tumors and neurologic complications of cancer therapy. His research activities include clinical, translational, and basic research in the fields of brain tumor biology, neurotoxicity, and brain repair mechanisms. He is the author of over 100 publications, including original research articles, review papers, book chapters, and other scientific contributions. His work has been supported by the NIH, the American Cancer Society, and the American Academy of Neurology. With that said, please join me in welcoming Dr. Dietrich.
Hello, everyone. I'm thrilled, delighted, and honored to present today during your webinar and conference. And I think it will hopefully be a entertaining and educational session for everyone. Even so I don't see the audience, I'm happy to address and answer to the best of my knowledge any questions you might have following this presentation. So with that, I'm going to share my screen. And you should be able to see this. So the title of the presentation, as you heard, is dealing, or the talk is dealing with chemo brain and related matters, which is a topic dear to any oncologist and any cancer patient. Here are briefly my disclosures. None of them are relevant to today's presentation. And so here the brief overview of what to expect. I will outline briefly the clinical problem that consists of the types of symptoms that are seen and reported by patients with a condition that is resembling chemo brain. That will followed by the potential mechanisms that the current state of research and investigations have taught us to underlie such symptoms. And lastly, how increasing knowledge in this field has informed the improvement in patient management and perhaps even allowed us to help patients recover more efficiently from perceived deficits. So with that, the clinical problem, as you are probably well aware of, is very common in cancer patients, and that does include patients who are treated not only for brain tumors, but also for tumors and hematological malignancies that not directly affect the central nervous system. And in particularly common are symptoms of cognitive impairment that are seen in patients who are treated with brain-directed therapies that includes radiation, but it can also occur, as you are probably aware of, after chemotherapy regimens alone. So the classic symptoms are to live somehow in a fog, to not be able to multitask, to be forgetful, not attentive, to have some lack in the ability to express themselves compared to their prior abilities, typically indicated as word finding difficulties, and all of the above as combinations may occur. And what has been interesting is that the field of studying cognitive deficits and impairment in cancer patients is still fairly new compared to many other disciplines and fields of investigations in the cancer domain. That said, the field is approximately 10 to 15 years old, while symptoms have been known for decades. Often these were attributed to mood changes. You're just depressed, and I understand that you are anxious, and this is why you're exhibiting some of these symptoms. But then eventually a more sophisticated and rigorous research has identified that these symptoms are actually real. And we know about certain risk factors that occur and about the pattern and the dynamics of cognitive changes in diverse patient populations. For you as an overview, what are the most important domains that are affected? And here are the ones named on the background of the brain, and to the area of the brain they are located to for the most part, which is attention, concentration, processing speed, and reaction time to the frontal areas of the brain, the frontal lobe, with important connections to the orbital and temporal pathways, so orbital, temporal, and frontal regions that are relevant to maintain executive function, which is sort of the accumulation of important cognitive domains that allow you to have perfect executive function. And then the symptoms that I mentioned that are often related to memory and learning that are more closely related to temporal regions of the brain. And then perhaps not as frequently visual spatial issues that might occur, difficulties with pattern recognition or orientation, or not finding simply the way from A to B on a map or in an area that you're normally familiar with or should be. The prevalence is fairly high, but as you can see, there is a huge range, 20 to 80%, and that is thought to be because studies have looked at very different patient populations, have used different methodologies, and have used different strategies to study this particular phenomenon. So in the worst case scenario, it might affect 8 out of 10 patients. In the least concerning studies, it has only affected a minority of patients with varying outcomes. Where it gets really complicated is that most patients are not just how should I phrase this, a specimen as part of a study or a subject. They're very complex, have complex lives, have other issues to deal with. And I'm specifically talking about genetic factors that might play a role, the age that might play a role, underlying anxiety and mood disorders, maybe fatigue that has been existing prior to the diagnosis of a particular cancer. And then there are on a cellular level issues that are very different in each individual, such as perception of oxidative stress, issues on the cellular function. So how are the detoxifying factories and mechanisms in your body able to bounce off and compensate for insult and injuries that are inflicted by the tumor or treatments, specific treatments. And so the ones I will focus on are highlighted in red, which is the chemotherapy and the radiation issues that have been best studied to cause cognitive impairment in cancer patients. And that brings me to the first imaging findings. So imaging is one of these tools that oncologists have to see behind the curtain, to see what else is obvious other than blood tests. Imaging is an important domain for patient assessment. And you see here that 3 individual patients treated with drug combinations that are very commonly used nowadays, old school drugs. ARC is one for acute myeloid leukemia, methotrexate and ARC combinations for CML, frequently used in the past, or for lymphoma alone, intravenous methotrexate. And on the right upper corner, you see a normal brain in a similar pattern where you should not really identify any large areas with white signal. And that is exactly the case here. So we call this white matter injury, which is the insulating material of axons or nerve fibers that has been damaged as a consequence of treatment and will usually lead to effects of functional decline. The myelin is one of the key cellular components that are relevant to a normal brain function. And this really across the board applies to anything we do, may it be motor function, sensory function, higher cognitive abilities. Myelin is sort of the insulating sheets that allow proper nerve conduction to occur. And if myelin falls apart, then you are in trouble. And this is exemplified in this cartoon here, because it allows you this conduction of impulses that are occurring in a saltatory fashion to break down, and that really slows down entire brain function. So thinking becomes slow, and you may have just simply trouble and deficits to do what you intend to do. So what are the mechanisms? And this has often and long been a mystery to lots of experts until the first animal and preclinical, so experimental studies identified what is occurring. And one of the major obstacles to shed light on this issue of chemobrain was a long established dogma by Ramon Cajal, who won the Nobel Prize in the 20s for his seminal work on brain architecture, on brain physiology, but also stating that whatever we have as tool sets at time of birth is what we will retain. And from there on, there's only really decline and dying. And if there is insult in the context of stroke, other brain injury or degeneration, then repair is really not possible. And this dogma has dominated neurosciences up until the 70s and 80s. And only it was in the 90s when key work by a group out of the US and Canada identified that songbirds, as the key cardinal animal model, were able to learn new tunes and songs each season, and were extremely resilient to neuronal injury, and were in fact able to come up with new pathways. And this triggered really a avalanche of research in the 90s and 2000s. And then eventually in the mammalian system, so in rodents and in humans, it was shown that there are two areas in the brain that are highly critical for maintaining plasticity. And so plasticity is in this context meant to be to form new nerve cells throughout adult life. And these new nerve cells can be integrated in existing pathways, can replace dying neurons. And this also applies to non-nerve cells, which are called glia in the nervous system. And these glia cells have a critical function to maintain neurological performance. And I give you two very striking publication results here from previous studies now published back in 2000 in PNAS that identified that navigational skills rely heavily on one area of the brain, which is called the hippocampus, which is located in the temporal regions. And this hippocampal structure is important for learning, memory, and navigational skills. They looked at taxi drivers in London. This was before the time of smartphones and iPhones and GPS systems, and found that these taxi drivers that are able to navigate a complex city like London have more higher volume hippocampal structures than people who are not able to complete these tasks. And then if you stop doing this type of skill work, then hippocampal volume and structure may revert to a lower level. Another striking example of brain plasticity is if you learn a new task, and it's something we all can do. Here, the example of joggling, that's something that typically you don't know how to do, but under study conditions, individuals were shown to generate new cortical areas, gray matter, so-called, in parts of the brain that are relevant to joggle and visually orchestrate and organize these balls you might keep in the air. And that is a task you can learn, and imaging to show that you can actually, through physical activity, influence your own brain. That is something that is striking for me as a neuroscientist and eye-opening, because of many interventions that we offer may in reverse help the brain to restructure itself. So for you to remember, one thing out of this talk is that there are different cell types in the brain that all need to work together, like a family that is healthy and supports each other in times of need. You need each component, and one alone cannot do it. So if I have a drug that targets these astrocytes here in green, you will have equal trouble than if I have a drug that might damage neurons, or drugs that could interfere with these myelin-forming cells, the oligodendrocytes. And here's an example how this looks in culture. So we have ways of studying cell biology and impact of certain drugs in culture systems before going into a higher complex model systems, such as a rodent or even in humans. And these young cells called progenitors that can divide and self-renew. And as they age, they form myelin, here labeled in red with a marker GALC. They lose this capacity of being a progenitor or a stem cell and eventually become post-mitotic. That means they don't divide anymore. They have reached their mature state and now are able to maintain neurological function. And so we have learned years ago, over 15 years ago, that certain drugs in fact damage myelin structures. So this is a control corpus callosum, one of the richly densest myelin areas in the brain. And after 5-FU, which is a fluorouracil, an anti-metabolite used in various cancer populations, you see a thinning of the corpus callosum. And these myelin sheets on electron microscopy thin out and actually get lost and damaged over time. Confocal microscopy allows you to really pin out individual cells and to see which cell populations in the brain are at highest risk. And then lastly, imaging, as I mentioned to you, has provided a big deal of information of what happens over time and in specific brain regions. Here's an example of patients treated for a type of brain cancer called glioblastoma. And over just a very brief period of time, which is about 4 to 6 months, these patients have profound brain volume loss. These fluid-filled spaces in black enlarge, and both the volume as well as the gray and white matter diminish in volume. We have looked at other imaging options such as PET studies. You know what PET studies are as a patient. They look for hotspots in the body, looking for uptakes of sugar and how it's metabolized. So it's glucose PET. And what we have identified is that patients were treated with a specific compound called cisplatin, a platinum drug for lung cancer. They don't have any neurological issues or brain tumors, had profound impairment of brain metabolism. So the sugar metabolism in the brain suffered over prolonged periods of time, allowing you to think more globally that even a treatment that is offered for a specific organ, such as the lung, may adversely impact the brain function. And so to make things a little bit more complex, anything that could interfere with cognitive function is often related to each other. So sleep deprivation, as an example, will lead to endogenous stress and endogenous cortisol production that general stress does as well. Increased in cortisol level will interfere with cognitive function and diminish neurogenesis, which is the process of forming new neurons in the area that is relevant for memory and thinking. And the same applies to depression and behavioral changes that then can go hand in hand with cognitive impairment. So it's a puzzle that often is very difficult to entangle. And this slide really summarizes many of the mechanisms that have been studied and now understood over the past 1 to 2 decades, which is that chronic inflammation, as it can be seen after cancer treatments, and particularly radiation and high-dose chemotherapy drugs, or direct cellular toxicity, which I have mentioned earlier, in concert with oxidative stress, will cause activation of microglia, which are the immune cells in the brain, will cause myelin damage as a next layer of insult, impairment of the ability to form new nerve cells, and also interfere with vasculature, which is very, very important to maintain neurological function. And as a consequence, you have processes of neurodegeneration that can be kicked in gear. And that has really formulated a novel concept, which is that treatments, and that does not apply for everyone, even so what I'm telling you sounds like all bad news, but there is a good news hidden within, which I will get to in a moment. But the concept that cancer on its own might shift our trajectory of aging, and treatment of cancer might worsen that trajectory has been a very attractive model and hypothesis to explain things that have been observed in various cancer patients. So the yellow line essentially would be the trajectory of cognitive function throughout life, where we all lose cognitive abilities over time. This can be shifted in a cancer survivor, but in patients who might have specific treatments that are offending to the nervous system, this could be accelerated in its decline. And so here are some of the factors that are relevant and known to be offenders and known to be supporters of brain function. So radiation and chemotherapy has now well been studied, as well as chronic inflammation that can occur in individuals that are chronically stressed or sleep deprived. Neurodegenerative conditions can emerge, and then certainly genetic factors play a role, because one patient might be perfectly fine, frankly, and the other one with exactly the same disease, the same age, and the same treatment is more or less devastated by the outcome. We do know that factors like exercise, certain nutritional aspects, an enriched environment, which is bringing us back to this issue of taxi driving in London, traveling, being exposed to new environments, learning a new task, such as a language or joggling for that matter, will help and constitute enriched environment. Antidepressants, interestingly, that have the ability to increase and enrich the formation of new nerve cells. And then also genetic factors, that these are all factors that could help to be more resilient or recover from insult. So what management options and interventions are there? The first one is, which you have heard about, I'm sure, is cognitive rehabilitation. We've gone through treatment. You have noticed some brain fog, some deficits and impaired abilities to multitask. You have trouble with short-term memory, perhaps. Cognitive rehab has been one of these anchors, and that typically is triggered after a detailed cognitive analysis or neurocognitive assessment that really studies all of these domains that I mentioned earlier in much higher detail, to see where are the most relevant impairments and how can we help each individual patient. Exercise and enriched environment is perhaps the most sophisticated, studied in experimental model systems. A mouse might run easily a marathon a day in their running wheel per day, which is not what we are built for or can do, but running as a cardiovascular vascular exercise modus is able to restore some of these brain functions that are lost. So this is something that we have built in our clinical practice with recommendations to incorporate into treatments cardiovascular exercise regimens. Here's an example. It's never too late to get on the road and do at least your daily walks. We now know from numerous clinical studies that it increases longevity, that exercise is able to improve quality of life and sleep and reduces stress levels. This brings me to the oxidative stress concept in general. This is probably the new frontier. You will hear a lot about metabolism and immune system. And so metabolism is where a lot of these things merge through diet, for instance. Diet, in a sense, is the biggest medication you take on a daily basis. And we know all too little about how certain food components can influence oxidative levels, redox pathways in cellular structures that are under attack, and how we can use perhaps dietary interventions to bounce back from moments of injury. So how can we translate many of these concepts to the bedside? The obvious one is we minimize exposure to drugs, we minimize exposure to radiation, and so avoid whole brain radiation, for instance, that has been done prophylactically in many instances. Perhaps we don't need it. Perhaps we can wait and delay it. We could offer, if we need radiation, what's called hippocampal sparing. So you avoid areas of plasticity that are the generators of new nerve cells and allow patients perhaps to not exhibit these negative side effects from radiation down the road. Or we work on certain modalities, types of radiation, and certain regimens that could be better tolerated and associated with less side effects. So here's the rationale. This is how it looks in practice. You just really literally block out certain parts, like here the hippocampus structure, which is where neurogenesis occurs. And that has been studied and is now incorporated in many centers across the United States and worldwide. Medication interventions have been on the horizon and are heavily studied. The ones that are most commonly used are neurostimulants, so drugs that will help you to be more aware, help you to focus and be more attentive. These are the same drugs that we use in kids for attention deficit disorders. They can also be used in adults, in patients with features of chemo brain. Antioxidants, ideally taken in form of healthy foods. Erythropoietin, which is a bone marrow stimulant to help overcome anemia, as you probably are aware of. And anemia is often associated with profound fatigue, and that really suppresses brain function. Anti-inflammatory drugs and cytoprotective drugs that are more often used in patients with degenerative diseases such as Alzheimer's have also been used with mixed success in patients thought to have chemo brain. And here's just a snapshot of many ongoing clinical trials. These are the clinical trial numbers. This is pulled from clinicaltrials.gov, where you can get an interesting insight of what is really going on an investigational level for different types of tumors: breast cancer, metastases, lung cancer, brain cancer, any type. They're basket trials. So there is a lot of activity, which is encouraging. And where I see the areas of basic sciences merge closest to improved patient management is in the domains of exercise, nutrition, sleep, avoiding toxicity or minimizing through changes in certain regimens, and understanding genetic factors. We kind of would like to know who is at highest risk before going into certain treatments. And in these individuals, ideally, one could perhaps come up with alternative solutions and regimens. So a group of experts came together at the NCI, FDA, and NIH to really discuss this. And we have published this guideline paper this year, which is accessible to everyone, to really highlight areas in need of research and areas where we have made some breakthroughs and advances. It's beyond the scope and detail of this talk, but there is emerging literature that might be helpful for you as a patient or a family member. And that brings me really back to this pros and cons slide. How can we restore the neurogenic niche, so the parts of the brain that help us to be resilient as we age and as we counteract and compensate for toxins that are coming across throughout life. And it doesn't necessarily have to be chemotherapy or the cancer, but in this case, it is a major offender. And how can we restore this resilience in the brain? And that brings me to one of our most recent own research works, which has helped us to understand that the brain is not acting in isolation. The bone marrow function is important for a healthy brain function. And that has to do with the fact that many of these cells circulate throughout our body through the bloodstream and then home to organs in need. And so here, using an example where the brain was targeted by an injury model, and for us to study which cell types of the bone marrow can come to rescue, can come to what areas of the brain and where they can set sharp and how they mature and how they differentiate over time and how they compensate. And we have learned that certain factors in the blood can compensate for cognitive impairment in individuals affected by neurological decline. And here's the cartoon really that helps you understand that the brain has these important areas, the ventricular zone or hippocampus, that has stem cells. These form more neurons and myelin-forming cells that maintain brain function. In the context of injury, such as radiation or chemotherapy, these self-renewing cells are under attack. You have loss of neurons, loss of myelin and white matter damage. And in the context of bone marrow support and mobilizing certain cell types that then circulate to the brain, we can restore this neurogenic niche to then compensate and form more than expected nerve cells. And that is something we are about to launch in a clinical trial in individuals exposed to cranial radiation, to brain radiation. So with that, I think just want to emphasize the fact that not only you need many different cell types to get the job done, but you need different organ systems to talk to each other, such as the bone marrow talking to the brain and vice versa, in order to function as a human being with a high cognitive ability. And I would like certainly to highlight a little bit where the field is going. We have learned a big deal on a cellular level. What are model systems that allow us to understand which drugs are most critical for injury? How can brain repair be started and engaging and trigger neuroregeneration? And how does that translate into maintaining cognitive function? And obviously in patients, provide cognitive assessments longitudinally, use imaging or other biomarkers, blood biomarkers, to monitor patients very carefully for insults from treatment. And then eventually, as we hope, have protective strategies in place in anyone who was treated. I would like to thank a long list of former and current collaborators to get some of this work done that we have contributed over the past 15 years. And obviously our supporting institutions and agencies. And with that, I share my talk and presentation, and thank you very much for your attention, and I'm happy to answer any questions.
Thank you so much, Dr. Dietrich. That was wonderful. We do have a lot of questions. Some were submitted previously. The first question is from a patient who wants to know, are there any supplements that you recommend for patients to take after they have successfully completed treatment in order to balance the effects of aging and or treatment?
Yeah, a wonderful question. I think we do not have any clinically established guidelines, but from a scientific perspective, certain antioxidants make perfect sense. The ones that come to mind are drugs like N-acetylcysteine, which are drugs that help restore mechanisms that help to detoxify from insult. Not to bore you with too much medical terms, but it's glutathione that is a pathway that is relevant for us to bounce off toxic injuries. And N-acetylcysteine is one of these drugs that can help restore that function. In general, I would advise to bank more on healthy diets and foods that are much more readily absorbed than supplements in general, unless you are unable to eat certain foods or have allergies or are otherwise functionally not able to maintain that. But if you have the choice to get your hands on leafy greens and berries, that is the way to go. And if you can't, then perhaps supplementing with some vitamins or antioxidants is not a bad idea in general.
Thank you so much. This patient says that they are on erythropoietin and they want to make sure that they're maximizing their brain power. Do you have maybe 2 or 3 things that you would say in priority order would be the best things for them to try and address in their lives?
I think the erythropoietin obviously is targeting the red blood cell lineage. And so if you have a compensated anemia, the anemia has resolved, then I think that is the goal. And that cannot really be measured as sort of brain power function, but the level of fatigue that travels with anemia would then be compensated. We do sometimes offer our patients these neurostimulants that can be quite helpful in the fatigued patient. Drugs such as methylphenidate or modafinil that are used to compensate for that have been useful, but not always. There are clinical studies that showed mixed results. And so I would then revert back to my initial recommendation is fresh air, cardiovascular exercise, light meals are often working wonders to restore some of the brain power.
Thank you. This patient wants to know, they have taken to doing crossword puzzles and are doing, this is an elder patient who has MDS, but they've taken up crossword puzzles and are taking online classes and are using the app Duolingo in order to learn a new language. Do you think this is a good approach to increase or improve or even just maintain? Any thoughts on those activities?
I do. So I think in general, they are oversold in a sense of promising more that they really can offer. But if you have the option to build it into your daily routine, that also should be driven by daily walks or exercise as much as you can, or a stationary bike, or at minimum a walk, then I think they are complementary. I would not simply rely on saying I've done all I could by doing crossword puzzles. But I think if you like that type of activity, I think that is encouraged to do. But I think you can enhance the benefit of brain regeneration by throwing in a lot of physical exercise on top of that.
Thank you, Dr. Dietrich. You mentioned that there are some studies that are ongoing. How would you direct a patient to find out about what search features should they use on clinicaltrials.gov to find studies looking at the impact of these medications or treatments on bone marrow failure patients, and how do you recommend that they get connected to those studies?
Yeah, so I think the most straightforward way would be to talk to your local oncologist, so the one you trust the most, and they can help you to navigate the system. Clinicaltrials.gov can be very helpful to use that site, and you can enter certain keywords and search criteria to look for active studies, to look where they are conducted. You can make restrictions. I only want to have studies in, I don't know, a certain city in the United States or certain region, only for a certain medical condition. So you can play with that. But I think if you also talk to your local oncologist, that would be the most helpful to see if there's anything in your vicinity, in your city or town or in your region that might be of help.
Thank you, doctor. Our next question comes from a patient who is also an MDS patient, who is concerned that that there is some hereditary concerns about I'm trying to read this. Sorry. This patient is concerned that their adult children and grandchildren may also suffer from the same effects because they're experiencing them now as an active patient. Should they be concerned that this is a precursor of something to come for their family?
So usually not. I think most of these syndromes are not inheritable or genetic per se. What could be genetic is the susceptibility to certain injuries. So to give you an example, if I would offer methotrexate to a patient and I know that a specific factor, which is an enzyme that helps to detoxify from that drug, is not working well, this could run in families. So if all the family members were exposed to methotrexate, they all could have a similar adverse effect from it. But if the family members, the kids or grandkids, are not treated per se with the same drugs or have the same condition, then this is not something that runs in families. None of these I would be aware of.
Thank you so much. This patient reports that after going through 2 rounds of ATG, they are now looking at having to go to a stem cell transplant. They felt that after their first round of ATG, which happened in their 40s, that they bounced back quite quickly, that they had a good response, and unfortunately had a relapse. And that after the second round of ATG, they feel that they have experienced a significant loss in brain function. They are experiencing significant fog. And now that they've had this second failure and are going to transplant, they are absolutely terrified because they are still a working professional and want to do whatever they can to maintain their level of function. Do you have any suggestions here?
The only immediate suggestion I would have is to, if this has not been done yet, obtain a more detailed cognitive analysis by a neuropsychologist. It's about an hour and a half of study, and that helps a little bit to set the baseline. What are the areas of the brain function that are suffering the most? And that would make you susceptible to choosing a specific interventions such as cognitive rehab, targeting areas such as if you have lack of attention, for instance, to compensate for that and to maintain your professional life.
Thank you so much. This patient feels that as a 50-year-old patient with a bone marrow failure disease, that they probably have adult ADHD. They also want you to know that they have self-diagnosed this condition by watching TikTok. But that being said, they are interested in understanding the impact of a well-treated and essentially in remission bone marrow failure disease, adult onset ADHD. And if there's anything that they need to know, do think about as they proceed through the rest of their life.
Yeah, so I think knowing sometimes what's wrong is already very helpful, not to be in the unknown and be clear of what to expect. And as I said earlier in my presentation, certain drugs have been used with quite good success, and methylphenidate is one of them. It is not unreasonable to try such a drug, and you would find out within days if it's beneficial or not. There are alternative medications as well. That's something that is best addressed by your local oncologist. They should be familiar with this, or at least make a referral to a neuropsychologist to address issues of attention deficit. And treatment is often very similar than it is in children.
Thank you so much. We have gone through all of the questions that we had from the audience and that were previously submitted. So I want to take the opportunity to thank Dr. Dietrich for his sharing his time and expertise with us. If you'd like to watch this session again at a later time, it will be available on the conference platform in about an hour, which you can access by clicking on the name of this session and click on the replay or on demand tab in the platform hub. If we were not able to answer your question today, or you didn't submit it, you can send your question to us at help, HELP, at aamds.org, or you can call our toll-free helpline at 800-747-2820. You can also submit your question through our Facebook page. Be sure to visit the exhibit hall. Now that we have a little bit of extra time, we would invite you to go and meet the AAMDSIF staff in our virtual booth. We would also encourage you to visit all of our exhibit halls, and don't don't forget to maximize your points by clicking on all the links and checking everything out on the platform. On behalf of the Aplastic Anemia and MDS International Foundation, thank you for joining us today and making us your resource of choice for information on bone marrow failure diseases. The AAMDSIF Medical Advisory Board, Board of Directors and staff are here to help you and your family as we have for the last 25 years, or 37 years, excuse me. This concludes today's session. Thank you.